Experiment 8 in Chem 1310 is titled The Nature of Acids and Bases. And in this experiment, we're going to look at species that either generate hydronium, H3O+, or hydroxide, OH-, when dissolved in aqueous solution. And these are known commonly as acids and bases. So we're going to investigate acids and bases found in common household products and measure the pHs of common household products. We're going to look at the effect of dilution and acid or base strength on pH, and we're going to investigate pH indicators, specifically looking at the pH at which an indicator changes color and how the relationship between the acidity constant, the equilibrium constant for acid dissociation for a weak acid indicator, and the pH at which it changes color tells us something about the microscopic ratio of acid and conjugate base species in solution. Let's begin with a brief introduction to Bronsted acids and bases. Bronsted acids are defined as proton donors. And when we're talking about a Bronsted acid in aqueous solution, we're talking about donating a proton specifically to water. So the most important reaction of a Bronsted acid in water is the reaction with water itself. What happens here is the transfer of a proton from HA to the water, forming what's called the conjugate base of HA, A-. This is HA minus one proton, right? Missing one proton relative to the original species. And we're also going to form the conjugate acid of water, which is H3O+, also known as the hydronium ion. It's the formation of H3O+ that really defines HA as an acid. Note also that we haven't drawn this as a complete reaction. We've drawn reversible arrows here to indicate that the reaction may not go to completion. What this means is, especially in the case of weak acids, we can think about this using concepts and terminology of chemical equilibrium, and specifically we can look at an equilibrium constant for this type of reaction, and that's called Ka, the acidity constant, acid dissociation constant, or most often we just say Ka. A Bronsted base is defined as a species that accepts a proton. If we're talking about a Bronsted base acting in water, we're talking about accepting a proton from water. So for example, a Bronsted base B, which has a lone pair which can accept a proton, reacts with water when it's placed in aqueous solution, just like the acid reacts with water, to form, not necessarily completely, the conjugate acid of the base, Hb+, this is B with an additional proton, as well as the conjugate base of water, which is OH-, also known as hydroxide. And it's the formation of hydroxide in aqueous solution that really signals to us that B is a base. Here again, we've drawn reversible arrows to indicate that the reaction is not necessarily complete, and we can again talk about an equilibrium constant for the reaction of the base with water, and that's called Kb, or the basicity constant, or the base association constant. If we're looking at strong acids and bases, we tend to think of these reactions as complete and just use forward arrows. So a strong acid dissociates completely or reacts with water completely. A strong base similarly reacts with water completely, and instead of worrying about equilibrium, we tend to just use the tools of stoichiometry to do, for example, calculations to determine the hydroxide and hydronium concentrations of a solution of an acid or a base in water. Now, the two are related by a reaction known as the self-ionization or autoionization of water. And this just involves two water molecules, one acting as a base and one acting as an acid. The one that acts as a base gains an additional proton and forms the conjugate acid, H3O+, and the one that acts as an acid gives up a proton, forming the conjugate base, OH-. This is a reversible process. There is a K value associated with it. It's known as Kw. If we think about the equilibrium expression for this, it's equal to the product of the H3O+ and OH- concentrations at equilibrium. And the value of this at 25 degrees C, as you may be aware, is 1 times 10 to the negative 14th. So it's a very small K value, so this happens to a very small degree. The important point, though, is that the H3O+ concentration and the OH- concentration are related to each other. So we can, for example, measure the H3O+ concentration of a solution of an acid or a base in water and know immediately the OH- concentration once we have that value in hand. Because of that, we tend to measure H3O+ concentration and express it not as that concentration directly, but as a value called pH. The reason we use pH is because these hydronium ion concentrations tend to be really, really tiny, and so we're ending up with very, very small numbers. To bring those numbers back up into sort of a more human domain, we take the negative base 10 logarithm of that concentration, and that gives us the value pH. And so you should think of pH really as concentration. It's just a mathematical transformation, the base 10 logarithm times -1 of the H3O plus concentration. And from the pH then, we can deduce the hydroxide concentration. Ultimately, if we think back to the self-ionization of water, what we can say is that 14, the negative base 10 logarithm of Kw, is equal to pH plus pOH. So acidic or basic species, when dissolved in water, are going to cause the pH to adjust either down or up respectively from the neutral value of 7. And what we're going to do in the first part of the experiment is to look at a number of household products with pHs that are different from 7. And we're going to measure those pH values. The thing to realize here is that when the pH does not equal 7, there must be some acidic or basic ingredient within the household product that causes that pH to deviate from 7. What I want you to think about in making these measurements is the question of why. What is it about the function of each product that makes it, for example, advantageous to have a pH above or below 7? Why is it a plus for the product to be basic or acidic? In the second part of the experiment, we're going to look at strong versus weak acids and bases and their effects on pH. To think about this, let's keep up these equations showing how acids and bases behave in water and think about the difference between a strong acid and a weak acid. And I'll let you do the base case on your own. We talked about previously that strong acids dissociate or react completely with water. What this means is that all of the HA that we start with in solution is consumed to form A- and H3O+. So what we can do, for example, if we wanted to calculate the equilibrium H3O+ concentration, or the equilibrium pH, the pH after we've dissolved some concentration of HA in water, is to simply take that concentration, realize that it will be equal to the H3O+ concentration, and take the negative log of it, and there's our pH. That's for a strong acid. A weak acid is different and is considerably more complicated because now in the weak acid case the reaction is not complete. So we have to bring in concepts of equilibrium, looking for example at an ice table and calculating H3O+ using the equilibrium expression for acid dissociation. For now, the important point I want to make is that if you think about the difference between a forward arrow and these reverse arrows, you'll realize that the concentration of H3O+ for a strong acid at some concentration is going to be greater than the concentration of H3O+ for a weak acid at the same concentration. So for example, if I started with 1 molar HCl, 1 mole per liter of HCl, and I compared that to a solution of 1 molar acetic acid. HCl is a strong acid, acetic acid a weak acid, and so HCl is going to have a larger hydronium ion concentration than acetic acid will. Because of the negative sign that shows up in the definition of pH, this means that the pH of the HCl solution is going to be less than now the pH of the acetic acid solution. A similar thought process applies to the basic case. I'll let you work that one out on your own, but think about the effects on pH of strong versus weak bases as well. We're also going to examine in this experiment the effects of dilution on the pH of a solution. And so what we're going to do is a series of what are called serial dilutions, where we start, for example, with a 0.1 molar solution in a first test tube, and we take 2 milliliters of that 0.1 molar solution, let's call that A. We take 2 milliliters of that A solution and we add to it 18 milliliters of water to prepare a new solution, B, which has a concentration of 0.01 mole per liter. That's again 2 and 18, so it's a 1 to 10 dilution, right? 2 to a total volume of 20. And then we do that again to generate another new solution, C, by taking 2 milliliters of B and adding to that 18 milliliters of water. This gets us to a concentration of 0.001 moles per liter. And I think we even do this one more time. The important point is, with each dilution, we're lowering the concentration of the, for example, acid by a factor of 10. So say we were working, for example, with 0.1 molar HCl. With each dilution, we lower the concentration of HCl by a factor of 10. And the question is, what are the effects on the pH? Well, given what we've just talked about with the fact that strong acids dissociate completely, and we can use the tools of stoichiometry, and here's the definition of pH, you should be able to predict what the effect of dilution is going to be on the pH of an HCl solution in a quantitative sense pretty easily. We don't need to worry about equilibrium or any of those complications. We just need to think about really solution stoichiometry and the definition of pH. The weak acid case is somewhat more complicated because here again we need to consider this concentration, the 0.1, 0.01, 0.001, as an initial concentration and worry about the equilibrium expression and an ice table and things like this. Even so, however, you can think about this qualitatively, and in the pre-lab for this experiment you'll do a series of dilutions and examine how the pH changes. So what we can say, for example, about the acidic solutions is that as we add water, we're lowering the concentration of the acid, and that intuitively should lower the concentration of H3O plus in the solution. So that should drive the pH up as we dilute the solution. But how much it goes up with each dilution is going to depend on the strength of the acid or the base, and you'll see how this works in the pre-lab. In the final part of this experiment, we're going to investigate pH indicators. And a pH indicator is simply a species that changes color in response to a change in pH. Specifically, if we think about pH on a horizontal axis, for an indicator there is a critical pH at which the color of a solution containing the indicator changes from one color to another color. So for example, for phenolphthalein, the solution goes from colorless at relatively low pHs to pink at relatively high pHs. In this experiment, you're going to be measuring this pH at which a color change occurs for phenolphthalein and one other indicator. But the purpose of doing this is not just to find out this number. The point of determining this pH is to come to a conclusion about the relative amounts of the weak acid form and the conjugate base form of the indicator when it changes color. So what I don't think I've mentioned yet is that an indicator is composed of a weak acid that is a different color from its conjugate base. So if we look, for example, at the case of phenolphthalein, phenolphtalene's acidic form, HA, is colorless. Its basic form, A-, is pink. And so where the color change occurs, in theory, is the pH at which A- becomes greater than HA. That is, the A- concentration becomes greater than the HA concentration. And in fact, by writing the equilibrium expression for acid dissociation, in other words, the equilibrium expression associated with this Ka, and taking the negative logarithm of both sides, we can write an equation that helps us relate the pH at which a color change occurs to the pKa of the weak acid, which we can simply look up, and the ratio of these concentrations, A- to HA. In a perfect world, the color change would occur at the pH where this ratio was exactly equal to 1, right? Since that would be the point where the A- concentration is just overtaking the HA concentration. However, because of different brightness, quote unquote, of the two conjugates, HA and A-, and imperfections of our eye and imperfections in how we're changing the pH, that's not going to be the case in practice. And the purpose of this part of the experiment really is to measure this pH and then go from this pH value to this ratio, A- to HA. This ratio at the color change is typically somewhere between 10 to 1, which in our case would simply manifest itself as equal to 10, and 1 to 10, which would show up for us as 0.1. And the purpose of measuring this pH is to determine whether for your indicator this rule of thumb, quote unquote, that the ratio is between 10 to 1 and 1 to 10 actually holds true or not. So once you've measured the pH, look up the pKa value and then solve not for these concentrations individually, but for the entire ratio. And this is a common point of confusion with the indicators part of this experiment. What we're looking for is this whole ratio, not the individual concentrations. In fact, you won't know those because you'll be given a bottle, for example, of phenolphthalein indicator that may not even have a concentration written on it. In fact, we don't need that. We're not interested in that. We're interested in the entire ratio here.